During metaphase, the chromosomes then line up at the equator, also known as the metaphase plate. And yes, you could also talk about how it's comparing both animals and plant cells. They're both the same. Oh, you did it in Bureau Below. My bad. I haven't read it yet. I'm being such a classic examiner right now, just preteen before I have even read everything. Anaphase occurs next, where the sister chromatids separate at the centromere and are pulled to opposite poles. So they actually separate and they're pulled by the centromere. So that's just a little bit of detail there. By shortening the spindle fibers, and then when at the opposite poles, they begin to decondense, causing the last stage of telophase. Both animal and plant cells, all 4 stages take place. Well done. With the exception of animal cells using centrioles to form the spindle fibers, plants lack these structures. I love this. I love this answer. I love how it goes through similarities, explains the process, how it's both similar, but then tells you at the end, okay, that's the difference. Okay. Cytokinase is an additional process occurring after mitosis where division of cytoplasm occurs, forming 2 different cells. Perfect. Animal cells involve actin and myosin to form a contractile ring. Yes, well done. The cell membrane inches inwards, thus forming the cleavage furrow. Well done, which then splits the cell into 2. Perfect. Now, the opposite is vesicles from the Golgi move to the center, and they contain cellulose, and they fuse together to form the cell plate, which then go outwards to form the cell wall. So this containing cellulose, I'll just add that little bit of detail because those are the details that examiners are looking for. Okay, those are the details that examiners are looking for. So you have to say that it contains cellulose. Okay, I would add that part there. The other part is I would try to make your answer a bit more balanced, where you've talked a lot about the differences here, and I would just make it a bit more balanced by adding a bit more similarity because it's the compare and contrast. But like I said, after I read it, I saw that you actually talk about everything is the same except for this. Okay, and here you talk about cytokinase forming 2 different cells, and both animals and plant cells have cytokinesis. I would just add that statement to, again, just make it very clear to the examiner. So both animal and plant cells have cytokinesis or undergo cytokinesis to make it very, very clear. And animal cells, however, do this. So when you're trying to phrase your sentences, be like, this is both, however, this is different. Make it very clear because they are reading literally hundreds of papers, hundreds of questions, and they're marking hundreds and hundreds and hundreds. So what you want to do is make sure that your answers are very clear, exactly I'm structured, I know exactly what I'm doing, and I've made it balanced. So to make it super easy for the examiner to give you those marks. Okay, let's read the next one. Mitosis is where the division of the cell happens. So here, yes, it is cell division and where the body grows new cells and repairs itself. Perfect. So you've told me the function and you told me the process. Fine. And the division of the nucleus. Now for this answer, remember that the IB will give you 2 marks for the quality of construction. If you are getting 2 marks for quality of construction marks, you want to make sure that your answer is written out in full. So write full sentences rather than bullet points. But I know that for this for this specific session, it's fine because obviously you're just kind of going through what you're just going through things that will get you marks. Absolutely fine. Don't worry. Just for in terms of exams. Okay. Yeah, it was the fastest. Yeah. No worries. No worries whatsoever. But yeah, so that's just one thing to keep in mind, okay? So mitosis, and I would say another term that examiners love to see is instead of saying mitosis is where this happens, because then that kind of implies the location. Say when you are defining things, guys, top tip, I got this tip from my business teacher when I did IB, and I have been using it ever since. Whenever you're defining something, just say refers to. Refers to the process where cell division happens, or refers to the process of cell division. So refers to is such a good word to use in exams, not just in bio, but generally, because it sounds really professional. It's really simple and easy to remember. So use the word refers to, especially when you're defining things, defining processes, and you'll literally see your writing become much more clear and better because you're trying to use those words that examiners love to read, and it's just so easy. So use the refers to rather than saying where this happened, just to avoid making mistakes and you don't want to get marked down for your sentence structure. So mitosis refers to the division of the cell, and its purpose is to blah blah blah. And it's not where because where again implies the location. Say its purpose is it's for cell growth, for the body to grow, and cell repair. Well done. And it also refers to the division of the nucleus. So again, be very careful here. because what we're trying to do is divide the chromosomes to 2 cells rather than divide the nucleus, because actually during mitosis, remember, we have no nucleus. It's just chromosomes, okay? So it's actually the division of the, not nucleus, but the chromosomes between the daughter cells. Okay, so the chromosomes between the daughter cells. Cytokinesis is also part of cell division, but here is where the cytosplitting, the physical splitting of the cytoplasm happens. Well done. So for mitosis, the process is the same in plants and animals, PMAT, but in cytokinesis, the process for plants and animals is different. Well done. So that's a really good sentence here. And even I would say this before, actually, this is fine because you define both and then you do this. You know what, I'll keep it the exact same. In both, new cell boundaries form and separate the daughter cells. Exactly. So both are, I think what you're trying to say here, in both, two new daughter cells are created, or using both. Oh no, you mean in cytokinesis, right? Yeah, you mean in cytokinesis. So again, be a bit more clear so the examiner understands. So in both forms of cytokinesis, in both cells, be clear on what? In both of what? Okay, because that's exactly the type of question that the examiner asks. Of what? Have you guys seen that, like your teacher grading and she just like writes, or they just write question, where, question, what, of what, and it's like, you know what I'm talking about, like, you know what I'm talking about, right? But this is the kind of nitpicky thing that they're saying. So write out exactly everything specific. And this is where leaving that line in between your answer in your long answer question helps, because you can go back and read and think, have I left out anything? Is anything unclear? Is there anything that my teacher could go back and write question mark next to? Right? So then go back and fill in that extra bit of detail. And honestly, it helps so much because you have lots of paper to use. Also, both of them happen during the M phase. So you mean mitosis phase. So in this case, remember that mitosis happens, so prophase, metaphase, and telophase, and then cytokinesis happens as a separate stage. So that point would basically make the examiner think, whoa, do they understand the processes? Do they not understand the process? You can say both happen after DNA replication interphase. Absolutely, you can say that. But the M phase or the mitosis phase is prophase, metaphase, anaphase, telophase. Cytokinesis happens after that. And if you guys remember from the crash course, we had that cell cycle diagram, and that cell cycle diagram showed you that PMAT and then cytokinesis, okay? But I think you mean the cell division phase. So you mean the cell division phase of the cell cycle, okay? And again, phase of what? It's the phase of what? It's the phase of the cell cycle, okay? Again, being more clear, because imagine you say this phase, examiner's going to go question of what, right? So that's the kind of thing that we need to unlock our examiner brains and think, have I missed anything? Is any sentence, can I extend a sentence to add more detail? You know, can I complete my sentence by making the location of a process more clear? Can I make it more clear in any way? And that's when you're going to leave those spaces in between your lines and you're going to go back and add them in. Sometimes it's hard when you're writing to think about everything, and you can go back and add it in and it's fine, okay? Mitosis has specific stages and cytokinesis is more of a continuous process. Perfect. So mitosis has 4 stages and cytokinesis has 1. That's a compare statement, okay? So I like that this answer was balanced. I would just make it more into paragraph form, or at least full sentences like the others have done. So here we have similarities and differences. Now, this structure of similarities and differences is really good for your 4-mark questions, for your 3-mark questions, but for your 8-mark questions, you want to write out everything, okay? And have you asked here, do you need to go into detail about steps of mitosis? Actually, I would just do it. The reason why I would just do it is because of the fact that you never know what's going to end up on the mark scheme, okay? You literally never know. I'd rather you write more than write less and then miss out on marks, especially for those 8 markers, okay? If it's a 2-mark question, I'd be like, no, girl, it's fine. But because it's 8 marks, you literally never know what's going to end up on the mark scheme. And sometimes the randomest stuff shows up. Like, I'm not even kidding, like genuinely. So make sure you write every detail that you know, every single detail that you know, literally word vomit on the page. That's what I would recommend. Okay. So both result in identical daughter cells. Yes, so both actually are involved in the creation of identical daughter cells, right? Mitosis is the division of cells for growth and repair that results in 2 identical daughter cells. Well done. The daughter cells have a full set of chromosomes and it consists of the process of PMAT, and then you can go through it. Both occur in cell division. Yeah, so this is the part I was saying, similarities. Both are involved in cell division and both result in yes. So these statements I would put at the beginning and then say, however, these are some of the differences. Okay, and then also their aspect of comparing plant and animal cells, because in the question it says compare plant versus animal. You can tell me about how prophase is the same, metaphase is the same, anaphase is the same, And telophase is the same in your animal and plant cells, except for that formation of the spindle. The formation of the spindle that usually happens more with the centrioles in your animal cells, whereas with the plant cells don't really have that. That would be the one extra level of detail, the one extra level of pizzazz, you could say. And then in terms of cytokinesis, we know that, so we know that cytokinesis divides the cytoplasm of the cell and results in two identical cells with two nucleuses. Cytokinesis separates the cell organelles and the cytoplasm. Mitosis has 4 stages, So again, you've given me those differences. Whereas actually, again, I would prefer it if, for example, you just said, put it in paragraph form and give me some intro sentences about the two processes, how they're both in and then explain the difference. So you're going to tell me they're both involved in cell division, similarity, and then say, however, cytokinesis, da-da-da, and da-da-da. Then give me the similarity again. So this is how I would answer my question. Prophase, metaphase, anaphase, telophase is similar in both, in during mitosis. And then da-da-da, however, plant cells do this, animal cells do that. Great, so similarity, difference, similarity, difference, similarity, difference. And that's how you ensure that the examiner knows that you're giving me a balanced answer. Furthermore, use terms like both processes, and then full stop, and then however this. So again, the examiner is saying similarity, difference, similarity, difference, similarity, difference, okay? You can put all your similarities together and your differences together, but it's just harder to write about because then your structure gets a bit more disjointed because you're gonna talk about something and then you're gonna have to bring it up again to compare them, okay? So my advice, intro statement, what each process is, similarity, difference, for the compare and contrast. You keep your, exactly like an essay. This is kind of hard because I never think of these processes different in plant and animal. Also my answer because I rushed it. And yeah, don't worry, honestly, you will get so much more time. Can we add that plants do asexual reproduction through mitosis while animal cells use meiosis? Absolutely. Guys, whatever you're thinking of, if it's relevant, please add it in your paper because you don't know what's going to show up in the mark scheme. So have you guys been told before, like how the mark scheme works? Like how do examiners decide what goes in the mark scheme and what doesn't? Do you guys know how? Anyone can unmute, by the way. Yes, no, maybe so? No? Okay, so basically they literally sit in a room, or maybe online, I don't know. They literally sit in a room and they basically together and they go like, collectively, guys, what do we think is going to be put on the mark scheme? And then everyone says a point and then they go back and review again. They're like, okay, oh, actually, maybe I would have thought this about this question. And then they add it again. And because everyone thinks so differently, everyone is going to add different inputs and have interpreted the question differently. And that's why random things show up in the mark scheme. They'd have been like, I didn't get that from this question at all. And that's because they go back and add, okay? And then they'll do a second round where they're like, oh, actually, I read a few answers and a lot of this, and actually I do think it's valid. So I think we should add that to the mark scheme, okay? And that's why sometimes they release an initial mark scheme. I don't know if you guys have seen that. And then you see the final mark scheme. Oh, actually, we made a mistake. Actually, we'll accept this as well. Because they go through multiple rounds of moderation because they know how important this exam session is. Yeah, exactly. They're actually trying to be nice because it's actually better because then you can add more things and you're more likely to get marks. Okay, so yeah, that would be my overall advice for the different structures of your answers. And I'm glad that we went through a compare and contrast question today because yesterday we did a lot of discuss, actually we did a lot of explain questions, which are slightly different in their structure. Is that clear, guys? Or is it not clear? How are you guys finding that? Is that clear for this question? But it's really weird they also ask to compare plant and animal. Yes, so it is weird because basically with plant and animals, you want to talk about like the cell plate that forms with the vesicles and the cellulose for cytokinesis. You want to talk about in your prophase the formation of the spindle, how it's different in plants and animal cells. But then there's a lot of things that are the same because in plants and animals, they're both forms of equal cytokinesis. That's one thing I wanted to say for all of you guys to say is that similarities in cytokinesis is, yes, both result in two daughter cells, but actually in plants and animals, it's most likely like equal cytokinesis, except for in some animal situations, you get what? Unequal. And do you guys remember what example that is? Oogenesis. Okay, so sometimes you get unequal, and that's an oogenesis. But in plants, you will mostly get equal. Okay, so you can talk about the equal and unequal cytokinesis because that is a part that they've added to the syllabus. Okay, amazing, perfect. Right, so that would be that. So now let's look into the paper 1B questions, a bit more data interpretation, and these were ones that I found for genetics. So I'm going to give you guys a minute to type in your answers, And this is a 1-mark question. So it should be pretty simple to just put in literally one sentence. And we have 3 answers so far. I'm going to wait until maybe like 1 or 2 more people reply, just so we can go through it. But the question says, the table shows that the nuclear DNA content of some plant and animal species suggests giving a reason whether gametes or somatic cells have been used to provide the information. Okay. Now this kind of looks like a difficult question because you're like, how am I supposed to know if it's a gamete or a somatic cell? But you guys remember from the crash course what a gamete is and what a somatic cell is. So remember that the gamete is your egg and the sperm cells. Okay, these are the haploid number cells, and your diploid cells, or all the other cells in your body, are all somatic. Okay, so anything that's not a gamete is a somatic cell. So a somatic cell is the same thing as like any other cell in your body. And so those will have diploid numbers. So from these species, now I don't know what a honeybee has. I don't know what chromosome number that is, but I know what a human has. I know a human number is diploid, is 46. And so I know that they've collected cells with a diploid number of chromosomes. And so the answer is going to be somatic because we have the diploid number of chromosomes. Well done, anonymous Kiwi. Exactly. Okay, so somatic cells, because the chromosome numbers given are diploid. But can you guys specify in your answer chromosome number given for what? For what? That's what I would say. And it's exactly which species made you think that it was diploid? Homo sapiens, the humans, right? So the human species made you think it was diploid. So again, like exactly give me your whole thought process, because that is the best way to maximize marks. Okay, perfect. Next question. State one way in which chromosomes are classified. And yes, you guys ate this up. And remember that karyotype, that karyogram that we looked at, they organize it based off of size. Chromosome 1 is the largest. Chromosome 22 is the smallest. And then we have those sex chromosomes, those X and Y chromosomes. Well done. Perfect. Okay. Oh, you guys are doing amazing. You guys already answered everything. Outline the role of histones in plant and animal chromosomes. Now, again, a 1-mark question. So we're going to think of one thing to say. So histones are proteins that the DNA wraps around to form nucleosomes. Perfect. Helping to package and condense the DNA. Will also help regulate gene expression by controlling how tightly the DNA is wound. Wonderful. Perfect. And it's involved in what is known as DNA supercoiling, which is the condensing of the DNA. Now, anyone remember exactly what process, what the DNA supercoiling turns what into what? Remember, it turns your chromatin into chromosome. So you can even talk about that. So histones are the proteins that DNA is wrapped around, absolutely. But remember, it's the role of histones. So they actually don't want to hear about structure in this answer. I mean, you could put it by all means, but you have less space to write out this answer, and anything outside the box does not get accepted. So you have to make sure your answer fits in the box. So you don't have to talk about structure because it's a 1-mark and they've only asked about role. Furthermore, they've only said an outline. Outline means give me less detail. Give me little detail. You don't have to explain. You don't have to say something because of this, right? You can just outline. And the role is condensing the DNA and compact it into the structure. And it's not that it, yes, and this is fine, but it also, like, more importantly, is able to condense your DNA from your chromatin form, which is that loose form in your nucleolus, into your tightly wound chromosomal form, which we need during mitosis, during that cell division. Does that make sense, guys? So I would more think about how the DNA is condensed, the DNA is supercoiled, because of the fact that we need that for mitosis. Because if you have it in the chromatin form, it's super hard to divide amongst 2 cells. Okay? So that's why, basically. Okay, moving on to the next point. Sorry, moving on to the next point. Histones are proteins that present in eukaryotic cells, allow DNA to wrap around them, forming nucleosomes. This allows DNA to condense into chromosomes, and this is later helpful in prophase, which allows Again, guys, 1 more question. I know you guys want to show off your knowledge, and you guys are eating. Question, absolutely ate it up. You guys ate. However, I want you guys to, like, again, be realistic about how much you can fit into that statement and how much you actually need to say. And it's DNA supercoiling, or turning the chromatin into chromosomes, or condensing it. Exactly. Well done. Next one. Using the data provided in the table, discuss whether genome size positively correlates to organism complexity. Now, I specifically wanted to add into this session this question, because when it says the command term discuss, guys, you have to give a pro and a con to that hypothesis that they've said or whatever they've suggested. So here it says no, genome size does not positively correlate, and I appreciate that you guys are, and this is true, absolutely. However, you have to give both a supporting statement and a non-supporting statement. Because if you have a supporting statement and a non-supporting statement, that is what the command term discuss is asking for. It's not telling me suggest. If it said suggest, you guys can go whichever way you want. You can go pro, you can go con, you can do whatever you want, but it's discuss. And a discussion goes two ways. It goes positive and negative. So the easy thing that I want you, that everyone is going to have thought in this question, all the students would have said, no, it's definitely not correlating with genome complexity. Because remember, we said in the crash course, a very common trick that the IB puts into their questions is asking whether genome size correlates with complexity, right? And I like how you guys gave me answers, because that was the other point I was going to say. Some people here have even written exactly the species, who is more complex than the other, right? Who's more complex than the other. And I like that, because when you have a discuss statement, not only do you have to give pro and con, but you have to tell me the data. You have to tell me, back up your evidence and give me that evidence from that table. So two things you have to think of with discuss statements: give me evidence, and also pro and con. Very, very, very important. People on TikTok are saying that it's a bit stuck. I hope it has resolved. But anyways, yeah. What would the pro be? Yes, let's go through the pro. So let's go through the answers that you guys said, and we'll go through the pro, absolutely, because it is harder to think. Because the con is super simple, because we've seen this before, common IB trick. However, the pro is a bit more difficult. So the pro for this question would be looking at exactly what animals you have and what plants you have. So we know that a plant, we know that an animal, the animals here are the honeybees, the humans, the red panda, and we know that of these animals, who is the most complex? The most complex is number 46. Oh, people on TikTok are saying it's working again. Thanks. Thanks, guys, for letting us know. So the most complex organism is the 46 chromosome, the human. And so the pro would be, this is, out of the animals, the most complex is the human, and they actually have the largest genome size. That would be the pro. I know it's not great. I know it's not a very strong pro, but that is the pro for these questions. You have to think of, how can I manipulate the data to give a pro? And I know it's hard, but you just have to think of something. So these answers are great. However, they don't include the pro. Okay, so you want to make sure that you also include the pro. So I'm going to write it here. The pro is, yes. So anonymous Kiwi is saying, but doesn't the canopy plant have a larger number of genomes than maybe the human? And you're absolutely right. So, but again, you have to give a pro. You have to give a pro. And so we've given a con, which is the genome size is not positive questions, but the pro has to be something. And the strongest thing we can think of is that out of the animals, the most complex organism on there is the human, and they are the ones with the largest genome size. Okay, so I'll write it here. Out of the animals, caps lock makes me feel like I'm screaming. Out of the animals, the most complex is the human, and they have the largest genome size or chromosome number. Okay, is everyone on TikTok following with me as well? Largest genome or chromosome number. Okay, so your answers are good, and you're saying no, the genome size does not positively correlate to organism complexity. The red panda is a complex animal, which is also larger than the canopy plant, but the red panda has a smaller genome size. Thank you, guys. That is really, really good. And there is no positive correlation. So here, we're not going to talk about correlation, because we haven't seen a line graph. And I know you guys want to use that word because database questions is there. And I know that is a thing, but please don't use the word correlation unless you see a line graph, okay? If you see a line graph and two specific variables, then use correlation, okay? But you can say there is no relationship, because also here it's asking, there's no positive correlation. Oh, there's a question mentioned. Oh, no, fair enough, never mind. I just didn't read the question. If the question mentions correlation, or if there's a line graph, you can talk about that. But you don't have to say like, yeah, it says positive correlation. That's fine. My bad, guys. Ignore what I'm saying. There is no positive correlation between genome size and organism complexity. Perfect. Thank you, guys. Clock me on that. Ate it up. Because the genome size does not measure the number of genes of the complexity of the organism. So actually here, you don't have to explain why not. So that would be my feedback for you, whoever read this question. Don't explain why not, because in database questions, you're not asked to, unless it says deduce a possible reason or suggest a possible reason, then you got to turn your scientist brain on and try to think of a reason. But here, all you have to say is that actually there's no positive correlation between the genome size, and give me an example. Give me your evidence, give me your data from that question, and tell me actually the human is the most complex, but they don't have the most genomes, the greatest genome size. Okay, so that's what it is. Someone said, but saying human has the largest on TikTok is like incorrect because there are species with more bases, and I completely agree with you. But in database questions, you are restricted to the data they provide you in this question. You don't consider any other data. That's literally what it is. Okay, so that's why you have to kind of play the game. And I will show you the mark scheme, and I'll show you guys that you do have to say both pros and cons. And yes, and here I wouldn't include this, and instead I would change that statement to give examples. So my feedback for you would be to give an example from the data. So cite your data. So literally give me a comparison of two animals like this person here has. Okay, no genome size would not positively correlate to organism. Yes, this can be proven by comparing the huge genome of the canopy plant and the smaller one of a human. Thank you, you've given me evidence. Well, the number of genome is bigger compared to that of most complex of humans. A canopy less is a complex. Perfect. This is a perfect con, but guys, give me a pro, because in those discuss statements, I want discuss statements, and both pro and con, okay? And this is how you guys are going to get those 7s. So make sure that you write a pro and a con. That would be my advice for you. And let's have a look at the mark scheme, because I did have it out here so you guys can actually see exactly what they've written. And for question A, everyone got it right. Everyone talks about the fact that it was diploid, whatever. And we talked about how the chromosomes are organized by length, which is great. There are other things like the centromere position sometimes and the banding pattern, which provide them. But they, very importantly, they did not accept shape in that question. Correlation density of chromosomes, we talked about that. And here is that last one, that last question that we just said. So you want to say, yes, they may be positively correlated because humans may be the most complex animal species in the table and they have the largest genome. Absolutely. A pro, like we said. And then they might not be positively, sorry. I'm going to explain it with the con first, because that's the easier one. Reset, re-listen to my explanation, guys. Re-look at the question, answer. So it says, number one, the con, they are not positively correlated because actually the two plant species in that data table have a much larger genome than the animal species, but they are actually less complex, which is exactly what you guys said. You said that humans are the most complex, but they have a smaller genome than the other species plants. Okay, you could also have said that actually complexity is subjective and there's not enough data because there's other organisms that we have to consider. Any of those is fine. Now for the pro, you have to look at only your animal species. And I know Amelia in TikTok is saying, I mean, like the table provided has, I think, the canopy as the species with the most bases, and absolutely yes. But because it's so hard to find a pro, this is literally the pro, is you confine yourself to looking at only the animal species, and you look at who is the most complex amongst the animals. So it's possible that there's a positive correlation because in that table, amongst the animals only, so B, human, and something else, the humans are the most complex and they have the largest genome, only looking at the animals. You're taking a smaller snapshot. Does that make sense, guys? Can anyone unmute and let me know, or just put it onto the document as we're answering it? Whatever you like. But yeah, does that make sense? You're looking at, yeah? Perfect. Thank you so much. So yeah, that is literally what we're going to look at. Okay, so remember, pros and cons, discuss statements. Cool. So I hope that was helpful. Okay, everyone answered this question and said it was B. Well done. So what description is matched with the correct phase in meiosis 1? So guys, do you remember in meiosis 1, it's different to meiosis 2. If anyone knows, any crash course student, you guys are, I know you know this, and you can unmute if you want, or you can type it here in the document. So tell me the difference between the division of meiosis and the division of meiosis 2. Yes, anonymous Kiwi, go for it. So what is divided, what separates in meiosis 1 versus what separates of meiosis 2? So what is separating? Because this is a very 7-level distinction. Remember that in meiosis 1, it's the homologous chromosomes that are being separated. Yes, well done, Romy, well done, anonymous Kiwi. In meiosis 2, it's the sister chromatid separating. Very, 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 very important distinction there. Meiosis 1, it's the homologous chromosome separating, and meiosis 2 is the sister chromatid separating. Well done, guys. So absolutely. So in prophase 1, recombination occurs only between sister chromatids. That is not true, because in prophase 1, recombination occurs between homologous chromosomes, not sister chromatids. In metaphase 1, homologous chromosomes join together at each end of the cell. No, guys, they don't join at each end of the cell. That's completely different, and they don't even go to the opposite ends of the cell, and it's not in metaphase. Yeah, that's funny. And then anaphase 1, homologous chromosomes are pulled apart. So this does happen in anaphase 1, because remember, anaphase 1, homologous chromosomes separate. Anaphase 2, sister chromatids separate, okay? Very important distinction. And when you're writing those 8-mark questions, that is exactly the level of detail that you need to write, okay? Telophase 1, 2 diploid nuclei are produced. And it's true that you do produce in telophase 1 like 2 separate nuclei, but they're actually not diploid. I guess there are 2 diploid nucleus that are produced, but it's not nucleases. It's just divided. Exactly. What's the difference between mitosis and? So the answer is C, I think, guys. Right? Perfect. What is the difference between also meiosis and mitosis? That in meiosis, the homologous chromosome separates, not just the sister chromatids. Yes. So in mitosis, there's only one division. So in mitosis, it's only the sister chromatids separating. Okay. So the answer here is C. It's the homologous chromosomes being pulled apart in anaphase 1. In anaphase 2, it is the sister chromatids. And then in mitosis, it's just the sister chromatids. Thanks. Okay. Let's go. Question 8. So according to cell theory, living organisms are composed of cells, and I wanted you guys to talk about question B. Outline what occurs in cells in the first division of meiosis. We answered an 8-mark question on mitosis and cytokinesis. Now we're going to answer this 5-mark question on meiosis. So let's have a look at your answers. So first person here said meiosis 1 is the reduction division where the chromosome number reduces from 2N to haploid. So it's true that it's a reduction division, but remember, guys, that in meiosis 1, your chromosome number, it does reduce from 2N to haploid, but remember that in interphase before this, your 46 chromosomes have actually duplicated, and they became 92. Okay, so you're actually going from diploid to haploid, because it's always called diploid to haploid in cell division, but you're in a human, you're going from 92 chromosomes to 46. Okay, just that distinction, just to make sure that you guys are clear. Meiosis 1 chromosomes condense, and homologous pairs exchange genetic information through crossing over. Yes. Can you be more specific and tell me what process of meiosis 1, please? That would be perfect. That would be icing on the cake. So you could say prophase 1, the chromosomes condense, and the homologous pairs exchange genetic information. And guys, oh my God, I can see an old student in the chat. I'm so happy that you did really in your crash, and thanks to the crash course from M25. I'm so glad that you did. Okay, anyways. Prophase 1 chromosomes condense and homologous pairs exchange genetic information through crossing over. Well done. And just specify that it is, yes, you can say, yes, good question, Anonymous Q. You can say diploid to haploid. I just wanted you to make sure that you know that in humans, it's going from 92 to 46. But it's still called diploid to haploid. Every division is diploid to haploid. That's absolutely correct. But I'm just like, giving you that extra level of detail to help you out. So prophase 1 chromosomes condense and homologous chromosomes crossing over. Well done. And the nuclear membrane breaks down. So here, what an examiner might think is that you don't know the order. So you want to make sure that you write everything in the order. So in prophase 1, first of all, the nuclear membrane breaks down. Then the chromosomes condense, DNA supercoiling it's called, and then the homologous chromosomes pairs exchange genetic information through crossing over. And yes, Evan, in the chat, meiosis 1 is separation of homologous meiosis 2 is separation of sister chromatids. Well done. Metaphase 1, homologous chromosomes align along the metaphase plate. Thank you. Using correct vocabulary. I love it. Anaphase 1 is the spindle fibers pull the chromosomes to opposite poles of the cell. Exactly. And they contract. That's how they pull them to opposite. In telophase 1, the chromosomes arrive at the opposite poles of the cells and the nuclear membrane reforms. And then cytokinesis happens where the cytoplasm divides to split the cell into 2 haploid daughter cells. Well done, guys. That's exactly what happens. And remember that for a lot of questions, you can give an example. So I always like to end my 4 and 5 markers with, if you end your 4 and 5 markers and your 8 markers, or just add examples, guys, I'm not even kidding. Like so many times, 8 times out of 10 in that mark scheme, they will be an example. And this way, accept any example. So you can say, for example, especially with your chromosome numbers, to make sure that the examiner knows, I would add a statement here to say, for example, in humans, okay, in humans, what happens is interphase duplicates the chromosomes, so duplicates the DNA. So you have 92 chromosomes. After meiosis 1, each daughter cell, so each daughter cell will have 46 chromosomes. Okay, again, examples are key. They are genuinely like a hack for getting points on the mark scheme. Okay, so make sure you add an example. You can add here, you can add it at the whatever you like. In the first division of meiosis, homologous chromosomes are separate. Okay, let's go through this one, and then we'll go through that one. In the first division of meiosis, homologous chromosomes are separated to produce 2 haploid cells. We have prophase, where chromosomes condense and become visible. Yes, thank you. And they become visible again where? Visible to who? Okay, if you're going to add detail like that, you have to be very, very specific. So become visible under the microscope, maybe. Is that what you... And then homologous chromosomes pair up and they form the bivalent. This is a great word to use because bivalent is a key word, okay? And chromosomes condensing is also, and crossing over is another key word. So that's a very important thing that you guys can add into your papers is those keywords, bivalence, crossing over between non-sister chromatids, which then create genetic variation. Thank you. That's really, really good. And remember, it's crossing over between the non-sister chromatids, yes, but between the homologous chromosomes, okay? Then metaphase, where the bivalents line up at the equator of the cell. The spindle fibers will attach to each homologous chromosome from opposite poles. After that, the anaphase, where the homologous chromosomes apart to opposite poles, and the sister chromatids will stay attached to the centromere. Perfect. Lastly, telophase is where the chromosomes reach opposite poles and the cell divide by cytokinesis to form 2 haploid cells. Well done, guys. Very, 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 very good answer there. This is a perfect answer. Let's go through this one. Meiosis reduces the chromosome number by half. In prophase, homologous chromosomes pair up, one from the mother and one from the father, to form bivalence. Yes, it's called bivalent. This is where the crossing over occurs, which is the exchange of genetic material, which also leads to genetic variation. Yes, in metaphase, the bivalents line up at the equator of the cell and its independent assortment, so the orientation is random. I love this extra level of detail. Like this is like a 7 marker. This is a 7 marker, not even a 5 marker. This is where the spindle fibers start to attach to the chromosome, and you guys are doing so well, like making me proud. In anaphase 1, the homologous chromosomes separate as the spindle fibers pull each chromosome to the ends of opposite poles. Yes, in telophase, the chromosome reaches the poles and then the nuclear membrane reforms. Again, just adding that extra level of detail there. And in cytokinesis, divides the cell into 2 haploid cells where each chromosome still has 2 separate chromatids as they separate in anaphase 2. So just remember that each chromosome still has 2 separate chromatids. Again, say each homologous chromosome still has 2 sister chromatids, and they will later separate. Again, just making it more clear for the examiner. I mean, later separate it two during meiosis 2 or something like that. But these are great answers, guys, and you guys have done so well. Honestly, I'm so proud. Like, I can tell you guys were locked in during that. Like, I can tell. And hopefully this session was super useful. Anyone have any requests for any other question type? I did have another question that if you guys wanted to go through was this question about the different stages of cell cycle, but only if you have the time. So if anyone wants to unmute and let me know if you want a question or not, I'm also happy to not go through it.
Yeah, please do it.
Yeah, you want to do it? Okay. It's all 2 and 1 mark questions, but I think it's good because it helps you visualize. And the other thing I wanted to say for you guys could draw the cell cycle if you wanted to. And one thing I wanted to highlight, because I think point of confusion in the 8 mark questions, cell division is mitosis plus cytokinesis. It's very, very important. But cytokinesis is not part of mitosis. So if you in those 8 markers said cytokinesis is part of mitosis, but it's actually not. So cell division, of the portion of cell division of your cell cycle, is composed of PMAT plus cytokinesis as a separate process. So mitosis is composed of 4 stages, but cytokinesis is its own stage within cell division. So when you guys are trying to describe that, you can say that both cytokinesis and mitosis form part of the cell division part of the cell cycle. However, mitosis happens first and consists of 4 stages, while cytokinesis happens after. Okay? So hopefully that is very, very clear. And let's go through this last question together in this session, and then we will call it a day. And thank you guys for following along and staying engaged. So let's go through question A. So it says state processes occurring during interphase. And I'm going to just put some tables for you guys to answer here. And you guys can go ahead and answer. So in option A, we're going to add a table, answer, and yeah, I'll do feedback here. So yeah, give me, not option A, but like question A. And then let's do B. I'll do the exact same thing for B. Actually, word B, we don't have to actually do that because it's just labeling. And then we'll do C, distinguish between the outcomes of a cell dividing either but mitosis or meiosis. Now, this is a distinguished command term, which we actually haven't gone through together yet. Let's go. And then we have D, the mitotic index is an important prognostic tool for predicting the response of cancer cells to chemotherapy. Outline how the mitotic index is calculated. Okay. And then that will be the last question we do together. Okay. Perfect. And then you guys can feed back to me. Let me know, do you like to do like kind of a good mix of all the different question types, database and non-database? Do you want a session where it's just database questions, or are you happy like integrating them into the topics? You guys let me know in the Discord. If you guys let me know, and we have anonymous Kiwi writing here. Anyone else want to answer question A? Please feel free. It's a good opportunity. But yeah, it's asking which process occurring during interphase. Now your state command term is very easy. You can literally like word, comma, or very short bullet points. This is a really, really easy and really good command term. So your state command term, state the process occurring during interphase, is a very, very good command term. And it's a really nice one because you can just literally statement, comma, statement, comma, statement, comma. Okay, so absolutely in G1 phase, we have the cell. Guys, what is happening during G1? I want that level of detail pushing you, okay? I want that level of detail. And what's happening during G1 and G2 phase is that the cell is duplicating everything that is not DNA. So it's duplicating the number of organelles. It's duplicating the mitochondria, the cytoplasm, okay? All of those organelles. So it's growing and duplicating the organelles because it's getting ready for cell division, okay? And then G2 phase is the same. So it's the same thing as G1, but part 2. And S phase, absolutely smashed it. It's protein synthesis or DNA replication. Well done. And here we've got some more answers. Cell duplicates all the organelles and grows, exactly, and grows. G2 is absolutely the same. And during S phase, we have protein synthesis and DNA replication. Should we also talk about the cyclins, the checkpoints? Someone asked that about in the chat on TikTok. You don't have to do it because, again, it's just what's happening during interphase. State the process. You can talk about cyclins, but you don't have to. It's just a 2-mark question. But if your brain goes that way, then go that way. That would be great. Is this a higher level question? No, it is SL. This is all SL questions, and we'll go through higher level sessions specifically next week. So mitosis results in genetically identical. Okay, people are answering C as well. G2 is the same as G1. It's just part 2. So cell duplicates all the organelles and grows as well. Same thing. Perfect. Next one, it says distinguish between the outcomes of a cell dividing either by or meiosis. This is a hard question because, again, conceptually it can be a little bit difficult, but I really appreciate you guys attempting to answer. So mitosis results in genetically identical daughter cells and diploid cells. Perfect. While meiosis results in genetically unique daughter cells and 4 times haploid cells. Well done. Can you guys tell me a bit more? Add some more detail to that. So when would you need mitosis versus when you need meiosis, for example? What kind of cells does meiosis create? Okay, all of those questions should be running through your head. What kind of everything, right? So in meiosis, we're creating what? Meiosis, we are creating the gametes, which are then used for sex reproduction. Yes, well done, Romy. Well done, Anonymous Fox. Okay, and meiosis, for example, is if you're higher level, is your oogenesis, your spermatogenesis. You don't have to worry about this if you're SL, but you have to know that it creates those gametes. Well done in the chat and TikTok as well. Gametes, exactly. Okay, perfect. And let's look at how many marks. It is 2 marks, so we don't have to talk about it too much, but it's important that we talk about exactly what kind of cells are being created. Okay, exactly, perfect. Well done, guys. Results into 2 identical genetically meiosis results in 4 genetically different daughter cells. Yes, and exactly. And the reason why it's genetically different is because meiosis has that prophase 1 phase, which the crossing over takes place. And yes, good job in the chat. Mitosis is genetically identical. for example. And people are already answering D, well done. So mitotic index, it's the number of cells undergoing mitosis, absolutely, divided by the total number of cells undergoing plus interphase. Yes, I love that you guys made that distinction. And how do you calculate it, guys? You have to look at what cells that have what visible under the microscope. Cells that have chromosomes. Okay, that's what you're looking at. Okay, and how do you know that cells have chromosomes? Well, you can see them because they stain under the microscope. So again, another level of detail that's the kind of stuff that shows up in the mark scheme. It's just like everything surrounding that topic, even though it's not directly related. Sometimes it comes up, but this was a 1 mark question exactly. So already like stating the equation, you guys are literally fine. But it's just make sure here that it's not just cell division; it's total number of cells under the whole microscope. So division plus interphase as well. Okay, so in your denominator, it's division, a cell division plus interphase. Okay. All right, and that was the last question of today. I hope you guys found the session useful. Please let me know if you guys prefer like answering the questions live, or if you what I want, I guess, for you not to see the questions before I send them out. But would you guys prefer to see the questions before the session, answer them before the session, and then we just go through the answers? Or do you like the style of like answering it live? Let me know either way. Isn't it the ones that undergo mitosis for the index over the and yes, if you come SL, how do I start papers? I would honestly just start learning the content, going through the syllabus guide. I mean, I don't want to, you can maybe join the crash course or something. We do go through the whole syllabus from start to finish through SL and HL. Okay, guys, but that is the end of the session. For those of you who joined on Discord, thank you so much. I hope it was helpful. And please don't forget to fill out our feedback form because we really appreciate it. And yeah, thank you guys so much. Any questions from me? Please do let me know. No, we're not doing another crash course closer to exam time, but everything is recorded and has already been uploaded on the hub. So for those asking in the chat, like you can just literally, you guys can just literally watch the recordings of the sessions that have already taken place, and you can watch them 1-2 times speed, so it's even faster as well. But yeah, thank you guys so much on Discord. Like really appreciated. Was it helpful? Was the session helpful? Was it good? Was it bad? Do you guys prefer to have the questions beforehand and answer them before, or did you like the style of answering them live in the?
I think it was very helpful, and maybe we can do both, where you give us questions and then we also answer them live.
Okay, yeah, definitely. And what topics did you want to see next?
Human physiology, like everything about like heart and lungs, and also like nerves and muscles, and maybe
Okay,
like
amazing.
cell respiration and photosynthesis as well.
Yes, absolutely. Okay, cell resp and photosynthesis is an important one because those are like really easy 8 markers. Neural signaling. Okay, we've got neural signaling from Seraphima. Absolutely. So we're kind of looking at a mix. And then did you guys also want a session where it was just just database questions? Did you like the tips on database questions? Did you already know how to solve the database questions before?
We do more database.
Yeah, absolutely. Because sometimes like it's just about knowing the command terms, right? And sometimes like teachers don't teach you the command terms, or like I don't know about you, but like my teacher sent me a list and was like, here it is, and then you never go through it and you don't have time and you've got like 6 subjects. So yeah, we'll do a dedicated database question session. Hopefully IB Docs is back up, so it makes it easier for me to make my handouts. But yeah, thank you guys so much for joining me. I hope this was helpful. I hope it was motivating and everything like that. And someone's asking, is it possible to go through the HL Chem syllabus in a week or 2 if I have very good foundations? Yes, it is possible. You just have to like work like 10 hours a day for like a few of those days. So yeah, it's honestly anything is possible. But yeah, thank you guys so much for joining me. I'm probably going to end the session here and probably going to do another session either over the weekend or on Monday. Would either of those days work for you guys? Would you join as well? And hopefully next time we have less tech issues because we actually can properly stream on TikTok like off the bat next time.
Weekend would be very good if it's possible.
Yeah? Okay. I'll try to do weekend then. Maybe like Saturday or Sunday. I'll see if I can do either. But yeah, next session I'm gonna decide if we're gonna do more human physiology or more like photosynthesis and cell signaling or database, because I think those are the most commonly requested ones that you guys So we'll try to do a session for each of those. And maybe one of those sessions we'll do like purely HLs because we have a lot of like purely HL content to go through, especially for cell signaling. And that's an HL topic mostly. But yeah, thank you guys so much for joining, and I will see you next week. Bye-bye. Is it heart and Krebs for the muscle motility? Oh yeah, muscle, yeah, human physiology. And yeah, that's already mentioned. Well done. Bye-bye, guys. I'll see you. Bye-bye.
Thank you.
Thank you for joining.
Bye bye.